But
Yes.
here we are always so careful to include the prefix, sir.
Yeah, that is our Indian problem. Indian problem is like that.
Yeah. No, so in a formal letter, I would like to include, but where I'm personally connected is different. Ramesh, you want to start? You want to ask sir to start? I don't know if others are.
Yes, sir. Sir, we can start, sir. Thank you.
Good afternoon, everybody. Some people have joined on Cisco WebEx and many have joined on YouTube platform, madam. Students have joined on YouTube platform because they find Cisco WebEx a little bit tricky. So many have joined on YouTube. Good afternoon, everybody. We have with us very inspiring teacher, Dr. Indrani Karunasagar, madam. In fact, I am proud to say that madam was my teacher during my BFSc and MFSc. On behalf of Karnataka Science and Technology Academy, warm welcome to you, madam.
Thank you.
Dr. Indrani Karunasagar, madam, is the Director, Projects and DST Tech, NITE University. He is also the head of FAO Reference Center for Antimicrobial Resistance and Aquaculture Biosecurity. He is also the member of Electronic Expert Group on Antimicrobial Use, World Organization for Animal Health. Also Director, UNESCO Resource Center for Biotechnology. Madam is also coordinator of MSc Marine Biotechnology, sponsored by DBT. Madam started her illustrious career as a lecturer in microbiology in 1976 in Kasturba Medical College. Madam then joined the College of Fisheries as assistant professor, and then she was promoted as professor and head in the Department of Microbiology, College of Fisheries, Veterinary Animal and Fisheries University, Bangalore. Then she is also a member of International Jury for UNESCO Prize for Research in Life Sciences. Madam is also chairperson for Research Committee in the Sri Devaraj Aras Academy of Higher Education. He is also a member of Task Force for Center of Excellence in the area of aquaculture and marine biotechnology. He also has served in many national and international committees, including FAO, WHO, Joint Expert Committee on Microbial Risk Assessment. Then she is also an expert in the FAO committee. Madam is also a task force member in the Ministry of Science and Technology, and also Indian Council of Agriculture Research. Madam has been awarded and recognized with many prizes and awards. Just a few of them to name. Asian Fisheries Society Award 2017, then Research and Academic Excellence Award, Hindustan University. As we were discussing, Madam is also a recipient of Sir M. Vishweswaraya Award for Excellence in Research and Lifetime Contribution to Science and Technology from Government of Karnataka in 2014. He is also a recipient of National Technology Development Award by DST in 2014. He is also a recipient of Eminence in Aquatic Virology National Award by Asia Pacific Association of Virologists in 2013. She is also a recipient of Dr. T.S. Vasundhara Memorial Oration Award in 2012 by DRDO Mysore. Dr. J.V. Bhatt Endowment Award in 2012 by Manipal University. Madam is also a recipient of very prestigious P.V.R. Pillai National Award by Indian Fisheries Forum in 2008, and Rafi Ahmad Kidwai Award of ICR in 2006, and another prestigious award, MS Swaminathan Award by Professional Fisheries Graduate Forum, Mumbai in 2009. And she is also a distinguished alumnus award by Kasturba Medical College. And I'm very proud to say that Madam is also a fellow of KSTA. Madam's research interests include aquaculture, biotechnology, biomolecules from invertebrates, fish and shellfish, and so many others. With this very brief introduction, I request Madam Karunasagar to take over and deliver our talk. I think there is some internet problem. Madam, over to you for your talk. This is just a very...
Yes. Thank you so much. I would now
brief introduction.
First of all, yes, I
To begin
thank
our-
the Karnataka Science and Technology Academy for this opportunity, and to Professor Das for so kindly agreeing to be there to moderate and to help me when I am in trouble during my presentation, being an expert that he is, and all the members of his officials of KSTA. Thank you. And I do hope we can make strides in Karnataka, at least in a small way, through inspirational stories of scientists who have been recognized globally and who are waiting to be recognized, maybe from India someday. So there are a lot of young people, we hope, who are listening, who through this story can see for themselves a future receiving a Nobel Prize one day. So, with these few words, I will now share the screen and begin my talk.
Dr. Ramesh sir, I think her mic is mute.
No, she will start.
Okay.
Hello, can you see my screen?
Yes, ma'am, we can see
yes,
the
yes,
screen.
yes.
Okay, okay. So I shall now, can I begin, Professor Das?
Yes, yes,
Ma'am,
ma'am.
but the presentation is not visible. Your that meeting screen only itself is
Yes,
visible.
your presentation is not there. Just upload the presentation. You have uploaded actually initially. Now, is it gone or?
Okay.
They
I
have
thought-
used, madam, you have chosen the screen share option. So now you have to open your presentation, ma'am. So it will come now.
Yeah. No. So first it is
Not this one, ma'am.
not yet come.
Delete.
KST officials, is she able to share?
Yes, ma'am. Yes, sir.
She's sharing. No, okay. Maybe internet slow.
They have not yet started, I think. Ma'am, the presentation is open, right, ma'am?
ಇಲ್ಲ ಉಮೇಶ್ ಮೇಡಮ್
Sir?
ಓಪನಿಂಗ್ ಸ್ಲೈಡ್ ಮಾತ್ರ ಇರೋದು.
Okay, sir. But ಇದು ನಮ್ದು ಇದು ಸರ್ ಬ್ಯಾನರ್ ಇದು.
ಅದೇ ಇದೆ.
Banner is there. Our banner is there. KSTA banner is there.
Yes, sir.
ಅಲ್ಲಿ ಮೋಸ್ಟ್ ಪ್ರಾಬಬ್ಲಿ ಇಂಟರ್ನೆಟ್ ಪ್ರಾಬ್ಲಮ್.
I think internet problem is there.
It's minimized, but we are not able to share it.
Sharing problem.
ಉಮೇಶ್
What a shame. Actually, otherwise, what I should do is I should send you the presentation. Is that okay?
I think
Ma'am,
that
yes, ma'am, that will also fine.
that is fine.
No, I think if somebody tells me the steps, actually my presentation is minimized and kept here because I don't know whether I have not done on WebEx. I've usually done through all other platforms. So I don't
Ma'am,
know
I will
where-
guide you. I will tell you the instructions now. click on the
Yeah.
share option now
Yeah, 1 minute.
at the
I'll go
bottom.
one by one. Share, yes. Then,
Then select, there are many options, file
Yeah.
or screen or presentation or other applications. You can
So
either choose
screen, huh? Okay.
screen or PowerPoint presentation application.
Select tab to share.
Either of the one will.
No, I can see on my screen, Chrome tab, window, entire screen. Webinar, WebEx webinar invitation, Nobel lecture series is there. And if I share that,
No,
no,
no, this is not the thing.
Unfortunate.
No, it is mail, it is going.
This tab is sharing your screen.
Yeah. No, no, not stop. Stop sharing. It will go off the meeting. Cancel, cancel, cancel.
I don't know, sir, actually, where, because,
Application. Windows.
Madam, you can send it. Extreme difficulty already. You just sent it one copy.
No, it's not showing up here.
okay, can I just send the mail to you, sir? Can I send
I
it?
think-
Ramesh, can I send it to you? Yeah?
I think officials, KST officials, I think madam can share, is it not? You can send it. She can send to you by email.
Connectivity.
Ma'am, you just sent it. You just sent the email
Here we are not able.
attachment.
I don't know, it keeps, there's something saying connecting, it's still not connecting, but it's not getting connected, but it's saying connecting. So I don't know if it's at my end or at your end that this thing is
We'll
not.
try with PPT only.
Madam, or if the presentation you can mail me right away, I will try to share it from my end here.
Custom from this chat. No, it's not there. It's only after we share. Ma'am will mail them.
Okay, I'll just send it by email to them straight.
Yes, that will be good.
I'll close now. No? Shall I close everything?
Cancel.
There is a share option button, ma'am, below
Yeah.
the screen window.
It comes, yeah, it says we can't display your shared content. Make sure you've allowed permission to share content
Oh.
and then
You are
try
allowed,
it.
ma'am. You are allowed. I think you are using Mac or iPad. Something
Yeah,
like
Mac. I'm using Mac.
Mac. At times it happens like that.
Okay.
So you have to click allow permission, that also. Then only you will be able to share from Mac.
You have to give the permission, I think. No?
No, we have given you, ma'am, all the privileges. You are the presenter now.
Okay, we will mail them. Ma'am.
So iMac settings will be there. So you need to give permission there. Otherwise, you can share presentation here, ma'am. I will share it.
Yeah, I think I'll do that because we are already losing time, no? So,
Yes, ma'am.
yeah. I'll just send it to you right away.
Okay, ma'am.
Compose.
I'll send it to KST Ramesh, no? It will go their office. We'll get it. Okay. No, I'll just reply to them, you know?
Okay.
Messenger. No? Wait 1 minute. KSTA. Okay, let's see.
Yeah, reply. Yes.
Attach. How do I attach? This one, no?
Yes. Open.
KSTA.
Yeah. Perfect.
It always comes up so well.
Yeah.
I feel sorry for young people who may. Okay?
Send. Send. It is okay. I'm not able to.
Mail sent to ksta.gok@gmail.com, right, ma'am?
Yeah, I've sent it to KSTA, yes.
Yes.
Okay. Really wonder why I'm not able to share here. Shall I come to share here and try?
We'll try.
Share.
Give a try, ma'am, one more time.
Yeah, I'm trying.
Screen. Select this one. A Windows.
Cisco WebEx
Yeah.
meetings, huh?
Yeah.
Huh?
Yes, ma'am. Ma'am, just try this one. Let's see if that works.
Okay.
Share.
That's okay.
This
I
window.
think.
One last try. This one. Share. Now you open your meeting. Ma'am.
Mine, huh?
Yeah.
In Mac, there will be accessibility and security and privacy options, ma'am. So in that privacy, Cisco WebEx meeting has to be checked. Yeah, your screen is visible now.
Okay. Now
Your
we will.
screen is visible now.
No,
Open your PowerPoint presentation, ma'am.
yeah, I'll do that just now. I have to open from what I attached to them. Just now. Huh?
Where is that?
mRNA.
Yes. Ask ma'am
Huh?
if they can see.
Oh. Can you see it now? No.
No, not yet.
Not yet.
KSTA WebEx is sharing a window, it says. What happens?
Your screen is visible, ma'am, but the PowerPoint presentation
Oh,
is
enter the panelist password, it says.
No,
So I will just paste. The problem with Mac, sometimes changes will not be saved. Leave. Huh?
Ma'am, I think you are logged in twice. I will share it from here, ma'am. Just
Yeah,
check
I
it.
know.
that will be easy.
Hmm.
Ma'am, you just go on narrating, huh? Just go on narrating, huh? You just come out here.
No, I am sharing from my end.
That is good actually. Ma'am can now describe and request for slide change.
Yes.
Hello, I can see the screen. So
Yes, ma'am. Yes, ma'am.
Yes, ma'am, you can continue from here only and ask for the next slide. So
yeah, I think that is
officials
best.
will change it. Please go ahead, ma'am.
Sure. Okay, so a very good afternoon and apologies to all those who are online for the initial glitches, as is always expected in the digital world, with all the advances. A topic that has been assigned today is on the development of effective mRNA vaccines against COVID-19. And since this topic was selected in view of the Nobel Award that was given to two scientists, it is in the fitness of things that I briefly tell you first about the Nobel laureates, briefly, because as and when I run through the slides, I will give in the context of what they did, something about them. Katalin Kariko and Drew Weissman, two brilliant people, two young people, and what they achieved is the story of the mRNA vaccine. Katalin Kariko, of course, was born in 1955. So when I look back and I think, oh, she's only 3 years younger than me, but she's done a whole lot of things. It makes me feel, yes, there are people who are so determined in what they are doing that they definitely deserve a Nobel Prize. On receiving her PhD from Szeged University in 1982 in Hungary, she continued with postdoctoral research at the Hungarian Academy of Sciences until 1985, then moved to the USA, the land of opportunities. But all along her career, She moved places, she moved jobs only because projects came to end. She did not have money to fund what she wanted to do, but her desire to pursue her heart's, I should say, interest was the driving force for whatever she did. And therefore, her getting Nobel Prize with Drew Weissman, whom she actually met at a photocopying center when she was working in the US. And upon, you just talk until your photocopying is completed, she chanced to find that here was someone who also had a passion working in an area where she was also very interested. So keeping the interest in mind, they decided to come together, and it is highly exciting to see that while Katalin Kariko is a biochemist, Drew Weissman was a microbiologist, immunologist, of course, and a microbiologist. So coming together of microbiology and biochemistry has always done wonders, and this time was no exception. And their work has been very fascinating, and a little bit about their work I shall talk about as we go on. Drew Weissman, the younger of the people who got the Nobel Prize between the two of them, is also a very ardent researcher, and how they found their way from institutions where they were working, from university to a private company, is a story of success. Can I have the next slide, please? Now, before we begin, this particular topic encompasses areas that need to be understood. And when I was given to understand by KSTA that there would be high school students, undergraduates, postgraduates, and of course, senior people. Very senior people would understand the depth of the topic that was supposed to be mentioned here. But when we talk about the undergraduates, I thought it's in the fitness of things to divide this topic to where I would be able to talk about mRNA independently, about coronavirus, the COVID-19 pandemic having been caused by SARS-CoV-2, and about vaccines. So there are three concepts to be independently spoken about before we talk about mRNA for COVID-19 as a vaccine, mRNA vaccine for COVID-19. So all cells have macromolecules that comprise nucleic acids, the lipids, proteins, and carbohydrates. May I have the next slide, please? We will, however, be focusing on the nucleic acids to understand more about the mRNA molecule that was at the bottom line of the discovery of vaccine. So nucleic acid itself comprises either the double-stranded deoxyribonucleic acid or the single-stranded ribonucleic acid. Of course, these information and all will be present in all biochemistry, microbiology, and every undergraduate science books in school, the PUC books as well. And therefore, I will not spend too much time, having lost some time in organizing as well. Can I have the next slide, please? Now, what are all the different types of mRNA? Because, RNA itself. Now, when we say mRNA vaccine, it really means there are other alphabets associated with it. So in a cell, the three major types that are generally spoken about are the ribosomal RNA, the transfer RNA, the messenger RNA. Transfer RNA comprises about 70% of the quantity of RNA, while the transfer RNA would be about maybe 20%, 20 to 25, and mRNA is the least in quantity, being about maximum 5 to 10%. There are, of course, others like the nuclear RNA, small nuclear RNA, small nucleolar RNA, depending on where they're all located and function from. The miRNA, siRNA, and then we have the INC-RNA. Can I have the next slide, please? So in the central dogma in molecular biology, now we know that DNA through transcription gives rise to RNA, and RNA to protein. In fact, I was fortunate enough some years ago to be at the Bangalore Bio when Dr. Sidney Brenner was addressing, and he added one more dimension to the central dogma when he said DNA produces RNA, RNA produces protein, and protein makes money. Well, we will see that that story of protein making money will come here as well. And the money that our friends got through the Nobel Prize is something the whole world knows about. Well, sometimes you have RNA alone, especially in viruses and all, which can be reverse transcribed, and then you have a complementary DNA that is made. Can I have the next slide? Okay, now about viruses itself. We know we went through the pandemic, and this pandemic with the coronavirus was new and yet not new. New because it became a pandemic. The deaths were very large numbers, which attracted world attention. But we also need to be informed ourselves that coronaviruses have always existed. They are a large family and cause illness in humans and other animals as well. Human coronaviruses have often been associated with mild form of cold, the common cold that we experience. Many of them, us would have had them sometime or the other. However, in about 2003, I think there was a very major viral respiratory illness caused by the coronavirus, at that time referred to as severe acute respiratory syndrome viral illness. And therefore, the virus was called as SARS-associated coronavirus or SARS-CoV. And until recently, when we had a second bout of the same, we started calling it SARS-CoV-2 because SARS-CoV had already appeared about 2003. But in between, we had the Middle East respiratory virus, which was also another coronavirus affecting, of course, people, but it was restrictive and didn't become a pandemic. Can I have the next slide, please? Now, human coronaviruses, if we look at the origin, their ecological reservoir are the bats. But bats are host to a large number, over 600 viruses. And it's not therefore surprising that coronaviruses were found. These coronaviruses were also found in bats. But why did it become such a serious issue? Because when virus jumps the species barrier to humans from another intermediate animal host, then it results in extremely serious illness. And for the COVID-19 caused by the SARS-CoV-2 virus, there is still debate going on in what has been the intermediate host. It could have been a domestic food animal, a wild animal, or a domesticated animal, which has not yet been identified. The search is still on and debates keep on going. Can I have the next slide, please? So coronaviruses, we need to know about the structure, because when we are going to see what structure of the virus was chosen for making a vaccine, we need to understand the virus itself. So they belong to the family Coronaviridae in the Nidovirilis family, the order rather, in the order Nidovirilis. And the name comes from the fact that it has crown-like spikes on the outer surface of the virus. The nucleic acid core, of course, it's an RNA virus, and the proteins are found on the exterior. These are enveloped viruses. That is because we have also viruses in affecting humans or animals, or present in nature, even innocuous viruses, or even bacteriophages that contain RNA. So you can have DNA or RNA viruses. They have only one nucleic acid, but coronaviruses are RNA viruses. And you can have naked or enveloped viruses. And these coronaviruses are enveloped with a size range of 65 to 125 nanometers dia. and they have a single-stranded nucleic acid as a nuclear material. Now, this again is very interesting because we are always taught that RNA is single-stranded. But in viruses, when we learn about classification, we have single and double-stranded RNA viruses. We have single and double-stranded DNA viruses. So it may not be usually taught in the very beginning when we teach about the strands of viruses, but it is good for us to know that the classification in virology is based on them being DNA or RNA viruses, being single or double-stranded in each case, deviating from the common definition of the strands of DNA and RNA. Can I have the next slide? So the virus that caused the epidemic was therefore called SARS-CoV-2 and first emerged in Wuhan, China in late 2019. The outbreak spread, of course, from China to all other parts of the world and became a medical emergency that had to be addressed by intergovernmental organizations like the World Health Organization on a war footing. Now, the common symptoms were fever, dry cough, and tiredness. Mild cases may be running nose or sore throat, but the fervor with which they would spread and resulting in serious illness, difficulties in breathing, ultimately organ failure or death in many, many cases became a cause of concern. And we also saw as a corollary how there was deficiency of oxygen leading to, political intervention and redressals in the form of setting up oxygen generation plants. Vaccines was also one approach that was very seriously being worked upon. And therefore, and the major focus was on how to prevent the spread of the virus because it had a propensity to spread very rapidly in the community, very rapidly. We know about the masks, but we also know how people have to maintain social distancing, hand washing, and we have heard how the virus could be from any inanimate surface or from the air spreading, and all factors of the environment contributing to the spread, and some countries witnessing more deaths than others. Can I have the next slide, please? Now, the third part. So I discussed about the nucleic acid, I discussed about the virus, because these are all basic to what we are going to see about the discovery and what led to the Nobel Prize being given to the scientists who discovered the vaccine. So now vaccine itself, we are very familiar with. It's a biological preparation providing immunity to any agent. Now, we are not at this time talking about bacteria or virus or fungus or any other form of life. What we are talking about is the ability to artificially bring about preparedness in an individual when there is a large volume of the pathogen affecting individuals. And if it is on a global scale, as in this case, becoming a pandemic, it naturally brought the world together to see what kind of protective measures could happen. The Nobel Prize went for one particular vaccine that was produced because of the history with which that particular agent was produced from, the incriminating agent. But there were other forms of vaccine also during this outbreak, where in India we had the killed vaccine being produced and Covaxin. We also had other types of vaccine that coming from the efforts of Oxford Group and and many other countries coming together to produce, okay? So as an example, if we see vaccination could be, of course, orally given, it could be through injectable, it could be also through and made by different methods. We could have a live vaccine which has been attenuated or a killed vaccine. Well, that is conventionally sometimes the most, I should say, accepted way of moving forward. And historically, if we see Edward Jenner saw that people who had ulcers in their hand, the milkmaids, were protected against pox as compared to others, and thought there must be something in the pustules developing that is giving them the protective immunity. And then they went on to also taking the spinal cord, drying it from a rabid dog, and injecting a boy who was bitten by a rabid dog and protecting. Well, ethics, if we start to talk, yes, they did not have to get as much ethical permission as we have to get today. But this was some of the beginnings of vaccine. And some of the vaccines that are very popular today, when we talk about live and killed, the best examples are the polio vaccine. You have the Salk vaccine and the Sabin vaccine. Salk is the killed vaccine, and Sabin, of course, it's after the scientist name, and Sabin is the live vaccine. So even in the case of, for the management of COVID-19, vaccine was produced in our laboratories, and we know from Bharat Biotech, the vaccine that was produced was found to be efficacious also in protecting. So conventionally, we know about live and killed vaccine, but we also know about recombinant protein vaccines as is taken for conditions such as herpes zoster infection and others. All of them are meant to elicit an immune response that helps protect against any serious illness. Can I have the next slide, please? So now, mRNA as a vaccine, I already mentioned mRNA, the messenger RNA as it's called, is found in the least quantity among the RNA, just 5 to 10%. And these vaccines again work by introducing a piece of RNA that corresponds to a viral protein. Now, when we talk about mRNA, we know that it is transcribed and then translated into a protein. So the mRNA could be a region that corresponds to a particular immune molecule. When I say immune, a molecule that can elicit immune response. So in this case, the mRNA vaccine the work itself was by introducing a piece of messenger RNA that corresponded to a viral protein. And in the coronavirus, it was a spike protein that was found on the virus outer membrane. And therefore, I gave the structure of the virus in the very beginning, where I showed that the envelope has spikes on the surface, and those spikes are proteinaceous in nature. Now, the spike protein, or peplomar, which is structure of an enveloped virus, aids entry of the virus into the cell, and this is done by interacting with the host cell receptors. Now, mRNA that codes for spike proteins are usually targeted as vaccines, mRNA vaccines, which can be injected intramuscularly. So if you look at the image here, we have the virus, and coronavirus is the one you can just say, of course, it's a cartoon, it's just a figure, okay? But this is how the virus is, that it is very beautiful and dancing. We saw in all the videos that were being displayed during the outbreak. All the time it was floating in the air, very beautifully covered with all those spikes on the surface like lovely flowers with the petals. So the spike protein, which is on the outside, is actually conjugated, and we can have any liposome or we can, a nanoparticle with which it is conjugated. And then we have it introduced into a human cell, where the translation of that messenger RNA coding for a protein, in this case, the specific spike protein, which comes to lie on the surface of the cell. And we know the human body or in animals, you have the antigen presenting cell. So in this case, it is the antigen which comes on the surface and elicits an active immune response. So the antibodies that are formed can neutralize these spike proteins and consequently, of course, ready itself and stay in a state of alert when there is an infectious agent attacking the individual. Can I have the next slide, please? So now the mechanism of the mRNA vaccine administration and its effects. See, the intramuscular injection of the messenger RNA that encodes the spike protein is taken up by cells in the body, and the spike protein is synthesized. The immune cells, which I mentioned earlier, the antigen presenting cells, they recognize these spike proteins as being foreign, or in other words, we call it as non-self, and present it to other immune cells. So while we are sensitizing a lot of cells, some of the cells also stay as memory cells to have an anamnestic response come about rapidly and in quantities much larger than the first time when you're vaccinated, which is why you have boosters of vaccinations being given. We have all been vaccinated. The mRNA vaccine also, they give every year the booster because some memory cells will be staying there. Now, the B cell produces specific antibodies against the antigen. It also produces memory cells that aids the rapid and enhanced response during the following encounter. Now, what are the merits and demerits of the mRNA vaccine? Looking at the merits, one thing is the rapid research and development. Today, we can say a simple production process, but the journey in identifying and producing it was very long, which is why the Nobel Prize goes to two scientists who worked on it tirelessly for 3 decades, not knowing they were working on a coronavirus vaccine, but they were working on an mRNA vaccine, not only for coronavirus, but for many other malignant conditions, for inherited disease conditions, and also conditions where proteins were responsible for the pathological effects. But in the case of corona, once they started there, when it occurred, people spoke of an mRNA vaccine being readied within 3 months of outbreak. But it also gave rise to people wondering how it was produced so rapidly. But it was a technique that was perfected rather than a vaccine that was perfected for the purpose for which it became extremely useful. So mRNA research was very, very, I should say, wonderful research and definitely deserved Nobel Prize. But its demerit is mRNA is unstable and easily degradable, and therefore we need to have it all introduced and prepared in a form where it will be readily available. And for all of this, we need the entire sequence of any virus or bacteria or a cancerous condition for which it is planned to be used. In the case of the COVID-19 outbreak, we know it occurred in around 2019, but I think within 2 to 3 months, the sequence of the virus was released from China. Now that was enough for the scientists to immediately start developing on the spike protein, conjugate it with liposome nanoparticles, inject it, and then have it ready at the very early period of the pandemic itself. Now, mRNA vaccines do not require nuclear localization signals and transcription in vivo. So one of the major steps in what the Nobel laureates did was in vitro transcribed mRNA. So that was also a major milestone before a vaccine was readied. Now, the molecule, the spike protein, of course, had strong immunogenicity. It was a demerit, triggering unnecessary immune response. Well, we are aware that when individuals were affected by coronavirus during the pandemic, many of them succumbed due to violent reaction, hyperimmune reaction, where a very large number of cytokines were produced, an extremely strong inflammatory response. So a natural infection could not have been thought of as being one that would immunize an individual, but there had to be steps taken to prepare a vaccine which would be different from merely looking at it as a natural infective virus that would afford immunity. Well, we'll come to that in some time from now, because the death that came due to the strong inflammatory response was a challenge to the Nobel laureates who had to see that they get a vaccine, but they do not get an inflammatory response that results in death of individuals. Now, the third important merit was there was no risk of integration of the material into the host DNA. Safety, of course, was lower than inactivated vaccines, going by the previous reasoning, but its effectiveness was found to be much higher than inactivated vaccine. Well, we all in India, through the Bharat Biotech vaccine, were introduced to the inactivated vaccine, which was believed to not be as effective as the mRNA. But we were happier that we would be saved of the extreme immune response with inflammatory molecules that most often resulted in death of individuals. But if we look at the demerit, the effectiveness is lower than DNA vaccines. Can I have the next slide, please? So I'm just going to run through how the vaccine was produced. It's very, very, very basic, okay? So the spike protein is the molecule that is immunogenic. So the mRNA coding for the spike protein has the instructions. Now, this mRNA portion from the virus was taken because we already have the full sequence. It was taken for making the spike protein and developed in the laboratory. So you can see everything was in vitro. And then this mRNA, which was of course conjugated, was made to enter a human cell where this COVID-19 virus spike protein was created to present itself on the surface of the cell. The spike proteins were recognized by the immune system as foreign and produced specific antibodies. And if one got infected with the virus, the antibodies bound to it and stopped it from replicating. Now, this is the very gross principle, and the two companies that were responsible for making it were the Pfizer, BioNTech. Pfizer was, of course, the one that joined hands, but BioNTech was the company where our Nobel laureates did a lot of their work. And the Moderna company also made the same vaccine. It was possible because they had the sequence of the virus. Can I have the next slide, please? Now, there are other COVID-19 vaccines also have to be briefly mentioned to you on how they are made. So material from the virus, in the case of, say, a vector vaccine. Now, all of us heard about the Oxford AstraZeneca vaccine, okay? Now, this Oxford AstraZeneca vaccine used an innocuous vector like adenovirus. So it has to be a vector that is not going to cause any other disease or problem, okay? It is really very carefully chosen. So material from the virus is placed in a different virus. So a viral vector, as we call it, is used, which gives the cell instructions to make copies of that protein. It's called S protein because it's the spike protein. Now, once the cells display the S protein on the surface, the immune system responds by creating antibodies and defensive white blood cells. We always have both humoral and cell-mediated immune response. So the picture says it all. And the vaccine was made by Johnson & Johnson. We also had the AstraZeneca vector vaccine. And the way it was produced is what you see in the picture here. Can I have the next slide, please? Then there are also this protein subunit vaccine, and that came about much later, the Novavax as it was called. Now, these subunit vaccines are only the part of a virus that give the best stimulation to the immune system to make the most protective antibodies. And this type of COVID vaccine contains, yes, spike protein, but they are, as I said, rid of their virulence potential. They are harmless. So once the immune system recognizes the spike protein, like before, they make antibodies and defensive white blood cell. And if one gets infected, then these antibodies fight the virus and the individual is protected. Can I have the next slide, please? Now we will come to the timelines that went into developing the mRNA vaccine, which is what got the scientists the Nobel Prize and which is the main topic. But without explaining all what I did until now, it would really not make too much sense to see the advantages and why they got the Nobel Prize. Okay, so it was in 1961 that this virus was discovered. Each of these milestones I will just discuss in each slide in the next few slides. So 1978, the isolated messenger RNA delivery into mammalian cells was standardized. The mRNA was synthesized in vitro, which was going to be very important. We cannot have only in vivo synthesis going on. And synthesis, and this is where it's very interesting to see that being a biochemist, Katalin Karika was a synthetic biochemist. She synthesized the molecules, actually, being a biochemist. And that helped a lot in what they were going to do later. The in vitro delivery was, of course, carried out around 1990. And in 2000, the in vitro transcribed messenger RNA, when injected to human cells, brought about intense inflammation. Now, the next step was how to prevent that inflammation. COVID had not yet happened. So we need to remember all this research went on before the pandemic struck and before the vaccine was brought to the market, before the necessity of a vaccine was felt. Researchers are in fact working for their passion, but also looking at the application to different fields in which they work, because what they work with is a technique. And today we see there are going to be a lot more applications of something that brought them Nobel Prize. So in 2000, we had the in vitro transcribed mRNA available, bringing up about inflammation. Now, how do we prevent the inflammation became a big question, even if it had to be used for other conditions like cancer or any other protein-related inherited diseases. So a nucleoside modification was done to make it non-immunogenic. When I say non-immunogenic, capable of eliciting immune response, but not the inflammatory response. And then came about the nanoparticle formulation, the lipid nanoparticle formulation, which resulted in the RNA vaccine that was prepared by the Moderna. And finally, it was in 2021 that both the companies got FDA approval for the mRNA vaccine. Can I have the next slide, please? So this is actually the beginnings of the discovery of mRNA from RNA. So the liable intermediate carrying the message from DNA to ribosome, it was not known before that. In fact, one of the questions with which Drew Weissman, the other Nobel laureate who won this prize, was intrigued, excited, wanting to find out was, how does information come from DNA to protein to ribosomes for making the protein? And it was that fascination that enabled him to study a lot about single-stranded molecules, with RNA being one of them and mRNA being the one that they researched most upon. Having said this, the references for all of these initial work that was published in 1961 by different authors, not necessarily Katalin or Drew, but many authors were involved at different stages of these cardinal discoveries. Can I have the next slide, please? Then the mRNA was now discovered, but in 1965, they, through various papers that are just listed here, the discovery of, as it is called, cap at the 5' end was done. The bases that are there at the 5' end, it's called as the messenger RNA caps. And the poly A at the 3' end, you have a poly A tail. When I say poly A, it's a repeat of A. So it's called a poly A tail. Now, these discoveries may sound very, very basic in nature to a person who works with applied sciences. And it is therefore at this juncture not out of place for a microbiologist like me, who is very fascinated with Louis Pasteur, the father of microbiology, who, when asked, when he was called by the academy, whether basic microbiology was important or applied microbiology, he only said, there is only one science, whether basic science or applied science. He said, there is only one science and the application of it. And this proves it even more. The discovery of the cap in the mRNA and the poly A tail were going to be the basis of every modification that happened for the mRNA vaccine that was generated, even to manage the big pandemic where millions of lives were lost. Can I have the next slide, please? Now, by 1978, we have this, the cap has been discovered, the poly A tail has been discovered. Now they intended to formulate the mRNA for its transfer into mammalian cells. Now that had to be done with the liposome. Today, nanoparticles have become a very important area of research, but we are talking about the beginnings of this research. So if you look at this slide, the rabbit leukocytes, reticulocytes, even before coming to human, the rabbit reticulocytes were taken. They are precursors of RBCs, as we know. The mRNA was isolated from them and the liposome formulation was done. So that was one of the important steps. And you can see, the extended portion of the mRNA with the cap and the poly A tail, which is shown there. I'm unfortunately not having the marker because I'm not operating it from my end, but I'm sure you can see it. You can see the P at the cap end, and you can see the AAA as the polytail. Now, the fusion of these cells with liposome was injected into mouse spleen lymphocytes. Upon this, the translation of rabbit globin mRNA introduced by liposomes into mouse lymphocytes was published and was one of the cardinal papers because immunoprecipitation showed a good, strong globin band. Can I have the next slide? Then soon thereafter came our heroine, Katalin Kariko, who actually studied the liposome-mediated DNA transfer into mammalian cells. As we know, we go through the stages of maybe a prokaryote to a eukaryote and then come to whatever has to be done with the mammalian cells. Having shown the success by the previous workers, in 1980s, she worked on formulating the transfer into mammalian cells. And the steps of the transfection experiments are also given in the next slide. Now, this she did before she moved to the US at the Institute of Biochemistry in Hungary at the university. Well, funding was a bit difficult and continuity in any research gets affected because funds are limited. And if people don't see the value of what you have done and look upon it as some basic research done in a lab of no value, then the projects don't get supported. In fact, the papers that were actually worked upon by Katalin and Drew were sent together to many journals, but they were rejected. And finally, when they thought they got exciting results and sent their publication, finally the journal Immunity accepted it. They thought the results were very exciting, but they failed to see the excitement in anybody else who read the paper. So you can have very, very negative impact when people do not appreciate what you have done. But this is also a lesson to all of us. What excites you in the form of having got exciting results may not excite others, but you never know along the path of research where what you have done finds an application that actually saves human race. Can I have the next slide, please? Yes, this is what was then done. I already mentioned how they were doing the in vitro synthesis, okay? So in 1984, the first in vitro synthesis of messenger RNA and the translation. See, synthesizing a nucleic acid is fine, but translating it into a protein in vitro is a challenge by itself. And what was done earlier in mouse lymphocytes and using, the reticulocyte, now was done in vitro, yes, in a test tube where the human interferon protein was taken and you had the plasmid DNA with the sp6 promoter. Now, the sp6 promoter itself is a very interesting promoter. Now, this particular molecule, of course, has many properties, which is why it was used for this purpose, okay? Now, the transcription capping was done, and with this polymerase and with the nucleotides and the capping enzymes, the process was achieved. The human interferon gene, and when micro-injected into the frog oocyte, what happened was striking. The human interferon was produced in 10 hours. So an in vitro method using a particular gene for a particular protein resulted in production of that protein through, of course, in vitro processes. And this led to them feeling confident that they could look at production of other important functional proteins that are responsible for a large number of diseases where they could control the problem itself. Can I have the next slide, please? So the synthesis of mRNA in a test tube and introduction to mammalian cells in the 80s was an exciting discovery, which I already mentioned. So the authors who did it parallelly, there were many scientists working on these kind of basic research. So the transfection was done into mammalian cells. That success was achieved. And the initial synthesis was in a test tube. Next slide, please. Now, the direct gene transfer into mouse muscle in vivo was the next step. And this was the in vitro mRNA that was synthesized and its delivery. Now, it is exciting to see the quantities of the protein that was getting produced when the mouse was injected, mouse muscle was injected. And so they were getting closer to what they wanted to actually work upon. Next slide. So this slide shows the in vitro transcribed mRNA, which was standardized by these scientists through the combined efforts of the two Nobel laureates, Katalin and Drew, was due to the in vitro transcribed mRNA being used and its expression. So in 1992, the vasopressin mRNA therapy was successful, and you can see them published in excellent journals. Then you had the nanoparticle mRNA that was made for influenza vaccine. Again, another mRNA for cancer vaccine in mice. So animal experiments, but I'm sure all of them are slowly seeing the light of the day for human intervention as well. mRNA for cancer vaccine in human trials also. Can I have the next one? All this with in vitro transcribed mRNA. So Drew now comes into the picture in a big way because he evaluated the GAG mRNA, GAG. Now, what does GAG stand for? Now, the antigen, this GAG is group antigen. And therefore, it was going to be really a little broad-based, but mainly for retroviral conditions. So you can see the HIV GAG mRNA transfection of dendritic cells delivers the encoded antigen to the major histocompatibility class 1 and class 2 molecules. Now, this was very, very important because the GAG viral assembly was going to enable the transfection of dendritic cells. Dendritic cell was only going to be a model to see how it is going to function. Can I have the next slide, please? So you can see the RNA, the transfection, and when introduced into the monocyte-derived dendritic cell, a violent inflammatory response to tumor necrosis factor alpha, TNF alpha, was observed. Now, though close to success, they were very disappointed because preparation of this kind of a molecule when injected to mouse resulted in death of all the animals. Well, it can be very disappointing, but we have heard of such stories even while studying the discovery of small interfering RNA, siRNA, how when it proved to be a great relief to solving almost all of the problems of humans through siRNA therapy, they found that when some animal experiments for some molecules were done, it had resulted in adverse reaction with all of the animals dying. So, as I mentioned earlier, curbing this major response, inflammatory response, was going to be a challenge for any of the conditions. Now, that being the case, can I have the next slide? How to prevent that inflammatory response? So, Carico and Weizmann noticed that cells recognize in vitro transcribed mRNA as a foreign substance, which leads to their activation and the release of inflammatory signaling molecules. And they began to wonder whether during translation, the RNA nucleotide bases of mammalian cells are frequently, generally, of course, they are modified chemically, but in vitro transcribed mRNA does not. So they wondered if altering the bases, absence of altered bases in the in vitro transcribed RNA could eliminate unwanted inflammatory reaction. So they knew what their task was. They wondered how to alter the bases. So they produced different variants of mRNA and each had a unique chemical alteration in their base and went on checking for inflammation. Lo and behold, they did chance to find some that completely did not have the inflammation or had reduced inflammation. Now, this really meant they were very close to success to what they were looking at, not for coronavirus vaccine, but for the large number of other clinical conditions that they thought they could manage with messenger RNA. Next slide, please. Here is the modification. So during 2005 to 2008, they modified uridine-containing mRNA and they found it to be non-immunogenic. When we use the word non-immunogenic, we really want to say that it did not produce the inflammatory reaction. So it was not hyperimmunogenic like the other cases. It translated the best but did not have the property of inflammatory responses. So the results of this study, where they altered it to have uridine, and from uridine to pseudouridine, made a big change. So if you look at the unmodified RNA and the base modified mRNA, you can see diagrammatically it has been shown how the inflammatory response was. So results of the study revealed that the inflammatory response could be abolished when base modifications are made to the mRNA. Next slide, please. So at this very time, they published another work which demonstrated that the delivery of chemically modified bases into the cell showed an increased protein production, reduction in unwanted inflammation compared to unmodified mRNA. They knew the race they were running was something where they were going to get success, and it was this technology which was used for the preparation of COVID-19 vaccine, and they were awarded the Nobel Prize in Physiology Medicine for this important discovery. Can I have the next slide? So if we look at the timeline from 1961, when RNA was discovered, and then the application of protamine on the RNA delivery, if we look at the interferon, which is inducible by mRNA, because I showed each of these experimental studies, the discovery of the phi cap, the polytail, the introduction of lipid materials, nanoparticles, liposomes to be used in mRNA delivery, or if we look at by 1994, the realization of in vitro transcription, RNA transcription, which was catalyzed by the sp6 RNA polymerase, the unique molecule. The discovery that cellular immunity is inducible, because we saw that both the defensive white cells as well as the antibodies were both responding to it. The discovery that humoral immunity was inducible, followed by, of course, the demonstration that modifying the nucleoside in the tail region, when I say nucleoside, we saw how uridine to pseudouridine did the job of making it non-immunogenic and producing reduced inflammation. And if we look at the first clinical trials ex vivo with the mRNA against cancer, which was published, and the utilization of direct injection of messenger RNA as a human cancer treatment, the first preclinical trials which delivered the targeted mRNA intranodally, the results with flu and respiratory syncytial virus, the influenza virus and the respiratory syncytial virus with nanoparticle and the specific in vitro transcribed RNA, the research trends all went to show that it was not surprising that in 2019, within 3 months of having the entire sequence, the phase 1 clinical trial was done. Within 3 months, it shocked the world, but they were successful. And then FDA approved the vaccine for its use in COVID-19. Next slide, please. So the statement from the people who really made a great contribution to the humans through this statement reveals a lot. They say 1 billion people have received the vaccine, but that's just one product. I see mRNA as being many different products for a variety of diseases and treatments of the future. Now, they have suffered a lot when they lost their ground in the US. I think BioNTech took them in Germany and they started to work there. And I think nothing stopped them from pursuing what they saw was going to be a success through their basic research that was being translated into possible treatment for a variety of diseases. Can I have the next slide, please? Yes, and that's the final slide. Dear Carrie Cohen Drew, many congratulations from all of us, from the whole world and from KSTA Thank you for your life-saving discoveries, not just for corona, but for a lot of other chronic conditions. With that, I say thank you very much to all the organizers.
Yes.
Madam, thank you very much for a very scholarly and informative talk on the works of Dr. Catalin Perico and Drew Wiesman. I'm sure many of the students who joined certainly would have understood a lot about virus and formation of vaccine. Right from basics, you explained applications, everything. It was really very nice talk, madam. I had forgotten everything about virus because of talk. I could recollect some information. On behalf of KSTA and all the participants, I am very grateful to you, madam, for your time and making it possible to deliver this talk. Thanks, madam. Now,
Thank you.
also, I'm thankful to Dr. Kushal Das sir, who has chaired this session. Now, over to you, sir, for your words.
Yeah, due to time, I would have interacted a lot if Madam would have been here, but there's a time restraint is also there. But I will only say the word that you have very leanly, smartly explained the functions and from generations to function of the COVID vaccine to the students keeping in the mind for just 17 to 18 years old. I think that is the grade. I hope that this will definitely go inside the students and they'll be motivated to become another, specifically the women to become another Carico, Dr. Carico. That is actually the world will get the benefit more. And that is the spirit which you have actually imbibed on the new generation of the student. The talk was excellent. And as a medical person, I will not comment because I was directly in the field of this corona. So I have seen, but this vaccine definitely has changed the entire story and their development. And that makes, and I also give credit to the government of India, the way it has handled, and many state government, they are so pioneer for implying and making the vaccination is very perfectful, which is US has failed. US people, many of them have, including Texas, they could not actually. I was in Texas just before the COVID. So they did not accept the COVID vaccine. Many French people also did not accept. Many other countries also not accept, but it make ultimately widely distributed. And so far, post-vaccination, any issues and concern, we are not getting. As a medical fraternity, we are not receiving any for the post-vaccination any issue except a 1-2 minor. There are no such report as that, which is the people were very much feared about the intake of the vaccination. That has gone. That is not there. Yes, post-COVID impact is still there. We are getting many of the issues in the medical science and clinical. Our doctors are getting a lot of that. But definitely, post-vaccination, any impact, badful, harmful impact is not there. It was a wrong conception have been imbibed by the many people in the globe. Your talk, your lectures, I think will inspire the young generations to pick up the microbiology or at least a virologist in future, that India will be benefited and my state will be very much benefited. And we are thankful to you. I thank Dr. Ramesh sir for giving me opportunity to be here and to interact with you. Thank you very much, ma'am.
Thank
Thanks.
you. Thank you. You are a COVID warrior, so we have to thank you for
Thank you. Thank you.
agreeing to be with all of us. And I'm honored that you have chaired the session.
No. It is my pleasure. Definitely, it's my pleasure. Thank you, ma'am. Thank you.
Thank
We have
you.
been joined by Dr. Hema madam. She is an eminent economist, Nobel laureate.
Good afternoon.
Good afternoon, madam. We are delayed by 10 minutes. Please excuse us.
No,
Good
that's fine. I caught the last 10 minutes of Professor's presentation.
But I'm sorry, I should have respect for the next speaker. And I lost
Thanks,
the first
madam.
10 minutes due to not being able
Umesh.
to.
No, that's no worries. Perfectly all right.
Thank you,
Umesh.
Dr. Hema, for giving me an opportunity to meet you as well.
So it will very
Invite online.
Meet you too, yeah.
Sorry, ma'am, there was a network issue.
No issues. Can I share my presentation?
Yes, ma'am. Yes, ma'am. Sir will introduce you, then we can start the presentation.
Okay, let me just. I hope Microsoft Cisco WebEx.
Due to interest of time, we will take the questions offline.
Sure. Perhaps I should email it.
Yeah, that's
Cisco.
fine, ma'am. When you click on the share option, ma'am, you will get many options like screen, application, or other
Yeah,
files.
I that's what I'm trying to do. It is saying I don't have system permission.
Oh,
to
you are
share.
using Mac, madam?
Yes, I'm using Mac. Is that a problem?
There will be some accessibility, and you have to go to privacy settings, then select WebEx application there.
Right. Then it says I will have to quit the contents and I have to quit and reopen. Shall I try that?
Yes, we can try that.
Okay. So give me a couple of minutes.
Yes, ma'am. Meanwhile, if any questions to Indrani,
In the
madam.
I'm also thankful to Dr. Akash and Dr. Philip who have joined for this webinar. Dr. Akash? Hello?
Dr. Idya Karun Sagar sir is also joined, sir.
Hello? Karunasagar sir.
Karunasagar sir.
ನೋಡಿ. Sir, ನಮಸ್ಕಾರ.
ನಮಸ್ಕಾರ.
Thanks for joining, sir.
ನಾನು ಮಲೇಷಿಯಾದಲ್ಲಿದ್ದೇನೆ. ಹಾಗೆ ಈಗ ಲೇಟ್ ಆಯ್ತು.
And we could understand so much about mRNA. In fact, I had forgotten microbiology. Madam's talk enabled me to recollect what he had taught. Thanks for joining, sir.
So now let me try and share.
Yes, madam.
Okay. Share Microsoft PowerPoint. Okay. Can you see my screen?
Yes, madam.
Let me try and make it full screen. And then does this work?
Done. It's okay.
All right.
So, madam, once again, welcome to the webinar. In fact, I'm very thankful to you. You readily accepted my invitation to deliver the talk on the work of Nobel laureate in economics. Madam will be speaking on women's labor market outcomes, for which Claudia Goldin has got the Nobel Prize in Economics. Before going to the talk, let me briefly introduce Madam's achievement. Dr. Hema Swaminathan is a professor at the Center for Public Policy in Indian Institute of Management, Bangalore. She is basically a BA Economics from Sophia College University, Mumbai. Then she pursued her MSc Economics from University of Bristol, UK. Then PhD in Agricultural Economics from Pennsylvania State University in 2003. Madam started her illustrious career in Indian Institute of Management. Before that, she has also worked in the International Center for Research on Women in the US for 3 years. She is also an economist. She has worked on poverty reduction and economic governance team. The Northeast Regional Center for Rural Development, Penn State, USA. She has also worked in Pennsylvania State University. She has also worked as a graduate assistant And Madam has received many awards and recognitions, including Visiting Scholar Program, Luxembourg in 2019. She has received many research grants. Awards include Shireen Latif Gender Operation Award for Best Gender Knowledge Product, and offered Bernard Berleson Postdoctoral Residential Fellowship and Population Council, then International Students and Scholars Grant in Aid Fellowship. Like this, Madam has received many fellowships and awards for her academic excellence. With this very brief introduction, Madam, I request you to take over and deliver your talk on women's labor market outcomes. Over to you, Madam.
Thank you very much, sir. Thank you for firstly, the invitation to give this lecture, and also thank you for your kind words of invitation. It is actually a very exciting time. I think we were all, many of us were really very happy when Claudia Golden got this Nobel Prize. I think it is many firsts, one of which is the fact that she is the sole recipient, and she is only the third recipient of the sort of so-called Nobel in economics. The last two were, of course, one was Esther Duflo with Abhijit Banerjee, and then previously it was Eleanor Ostrom for her influential work, along with a host of other people. So Claudia Golden getting it on her own, I think, represents a sort of a milestone. But also what is really very important is that she has gotten it for work, for a lifetime of work, studying gender in economics, right? So she's really, her lifetime of work has put women at the center of her research. And so it's, to that extent, I think people are very happy that there's been a belated recognition of the importance of the role that gender plays in economics. I mean, one can talk about it at length, but that's probably a topic for another day. So in terms of the way I have structured my talk today, is the outline. Briefly, I'll just walk you through it. Is that I'm going to talk a little bit about the motivation of why this is an important area of work, and why we should be thinking about it. And then I would like to very briefly introduce key aspects of Claudia Golden's work. And she's been active for more than 3 decades, so it's very hard to do justice to her body of research in this short span. But I will touch upon what I think are some seminal contributions of Golden's work. And then I also want to sort of draw it out and give an application of it to the Indian context to make it, I think, more relatable to our scholars, to our students who might be thinking about how this kind of work relates to, say, developing countries. The relations are very strong, and actually quite a bit of work has already been done. Her work has spawned quite a lot of research, both in the developed as well as the developing countries. Right. So with that, let me talk a little bit about the motivation. So my own research has also been a lot in the area of gender, and as was mentioned, thinking about poverty and inequality, right? So one of the motivations is that when we talk about gender inequality, and then we know that it is one of the SDG goals, goal 5, is that there's a very strong importance that is given to the economic aspects of gender inequality, right? And the reason for that is that economic empowerment, economic independence, and autonomy is considered very foundational to many other aspects of empowerment as well, right? If we talk about social empowerment, or we talk about agency, confidence, people's ability to sort of negotiate within the household, outside the household, economic empowerment becomes quite necessary, and it can be transformative. It's not the only form of empowerment out there, but it is a cornerstone when we start thinking about other resources, other kinds of confidence building and empowerment that can be given to women, right? And when we talk about women's labor market outcomes, it's important to also realize that a woman participating in a labor market, certainly it's about the job and it's about the earnings, but it's also about her ability then to build peer networks. It's about her ability to build social capital. It's about her ability to sort of step outside the household and sort of be a person in her own right, right? So it's about these kinds of issues as well that that are necessary when we start, that we should also be thinking about when we talk about women's labor market participation, right? So whenever we talk about gender inequality in labor market outcomes, a very common argument you will hear is that oh, women are not doing very well, or that there is a gender wage gap. It's because these are choices that women make. Or they don't go into fields that are high risk, or they are not interested in STEM. They do not have the ability to negotiate, or that their educational skills are much lower compared to men, right? But what you don't hear is that there are patterns of economic growth that drive certain occupational constraints for both men and women, but these constraints are particularly important for women, as I will talk later. And the fact that women's skill levels, their risk preferences, their ability to negotiate, and the choices that they make are not always exogenous, in the sense that these are not choices that women have made by themselves, right? When we talk gender, it's very important to know what gender socialization means, what boys and girls hear in societies throughout the world about what their roles and responsibilities are, right? So there is a certain social constructed identity and gender stereotypes of men's and women's roles in society. So when we say that these are choices that women have made, they have low educational levels, or they cannot negotiate, a lot of it is endogenous, in the sense that it's kind of within the system itself. And this is why it can also impact, right? So this is, and that there is what I call state-sanctioned discrimination that women face with respect to economic resources, right? So these are about laws and legislations and the institutional framework that matters, right? And you'll see that one of Claudia Goulden's key achievements is that she brought a unifying framework to understand women's labor market outcomes, many of which were pretty unheard of at that time. She really applied both a demand and a supply lens in her early work. And her later work actually started incorporating more of an institutional framework and thinking through norms of how these interact with women's labor market outcomes. Right. Very briefly, again, if you want to look for evidence of inequality in labor market outcomes, this is data from the World Bank Gender Data Portal. So all this tells you is labor force participation globally. So it's a female labor force participation as a percentage of the total labor force. You see that it's high in the darker numbers is much higher. So it's higher actually in Russia and the other CIS countries, or erstwhile USSR, Australia. It's much higher in Canada as compared to the US, and higher in certain other pockets of sub-Saharan Africa. Right? Very low, very light in India, and we'll talk about this when we come to the Indian context, and very light also in the MENA region, which is the Middle East and the North Africa region. Again, not surprising if we really think about the norms, the differential norms that play out there in these societies. Right? And further evidence of inequality in labor market outcomes is if you look at female labor income shares across the world. This data is notoriously hard to estimate because we don't have this data for a lot of countries systematically. So there's a lot of imputation that's been done here. This is from the World Inequality Report of 2022. And then what it shows is the darker colors where women are earning between 45 to 50% of the total labor income. So it's as low as 18% in India, which is a light blue, and it's the share of labor income earned by women was about 41% in France, which is amongst the higher countries. So in no countries it's actually greater than 50%. So in terms of the thinking about the key aspects of Golden's work is that I've kind of categorized it into 4 aspects. Her use of historical data and measurement of women's work, the importance of structural change that she highlighted, and then she also talked about the power of the pill, and then she talks about she has brought in some really interesting insights into the gender earnings gap. So broadly, this is what I'm going to touch upon. Of course, her work is more extensive and she has done other work, particularly earlier on in her dissertation years. And there are a whole body of papers. So I'm drawing from them. I've given you some of the big references towards the end of the presentation for those who are interested. But these are some critical aspects of Golden's work, and these are also the aspects that have been cited in the Nobel citation, and that the press release that follows the citation, that follows her announcement, right? So in terms of again, like I said, she was one of the, I think she probably was one of the first that provided a unifying framework for understanding women's labor market outcomes. She talked about the role of education. She looked at productivity. She looked at fertility issues, and she looked at institutional barriers. And then she also talked about the norms that dictate how men and women work and its contribution to the gender earnings gap. By institutional barriers, she was also looking at what are the kinds of constraints that married women face. I think even as late as the 1950s, there were what were called marriage bars. So women could not be hired, married women could not be hired by firms. So there was what I would call state sanctioned discrimination against women's participation in the formal labor market, right? And much of Golden's work is based on US labor market data. And she has done a bit of extension, but that also has been looking mainly at developed country data. But the bulk of her work is based on US labor market data. But like I said, this has spawned a lot of work and interest in other developed as well as developing countries. But her archival research was looking at US censuses and industrial censuses, as well as time use surveys and time reports, and those kinds of issues. Right. There have been some other people who've also looked at women's labor market outcomes, particularly in the context of developing countries. And it would be remiss not to mention Esther Boserup, who is a Danish economist who published her seminal work, Women's Role in Economic Development, in the 1970s. In fact, Golden herself refers to Esther Boserup's work. And Esther Boserup sort of really was very, very key in visibilizing women's work. Often it was thought that women don't do any work, particularly in agricultural systems, because they were not counted. But she did some very careful documentation, both in sub-Saharan Africa as well as in Asia, about the kind of work that women did and how this work was affected both by migration, by colonization, and population growth, and what are the impacts it has had in terms of thinking about women's empowerment, and whether they were male and female systems of farming, and whether women were out in the field doing work or they were more confined to the home, and how this has impacted empowerment. So we are trying, in fact, even today, people are trying to extend the insights from Esther Boserup's work in trying to understand how women's empowerment has spanned out globally. Just to say that Golden's work came a little later, and Golden herself talks about Esther Boserup's work and references it as such. Right. So in terms of the historical data and measurement, is that Golden did some pretty amazing, colossal and critical archival work that shows that women's work is estimated historically. right? And largely this was because of biases in definitions, changing definitions of work, and undercounting of women workers, right? And it's not very hard to think why this might happen, right? So in data collected before 1940s, women were often recorded as wives. That's it. Said, what does the woman in this household do? You know, or with a census enumerator, and the person who's responding says, "Oh, she's a homemaker," or "She's my wife," right? Which automatically implies that she's not engaged in economic in sort of active economic production, or she's not undertaking an economic activity, which therefore means that she is not counted as part of being in the labor force. But this was historically inaccurate, and it continues to be inaccurate, though we often still ask questions in these ways. And I'll come to that a little later. But women have always been active in terms of helping their husbands, helping their partners, supporting, sometimes even taking on their work, right? So wives of small farmers, tradesmen, boarding housekeepers, and other small businesses, they would work alongside their husbands. And you see some of this actually reflected if you pick up novels from those days. So if you pick up Jane Austen, or you pick up a Charles Dickens and you read it, you find that women are playing fairly economic roles, right? They do appear. They appear in literature, which is a reflection of society, but often they don't appear in national data systems, which is problematic because it's the data systems that actually drive our understanding of what men and women are doing, and we derive our policies from them, right? So Golden really dug deep into historical sources and showed that in 1890, she combined data from census, industrial policies, time use surveys, and a few other archival sources. And she showed that women's LFPR, according to her, rose by 7 percentage points. This is in the agricultural sector. And in the other smaller manufacturing sectors, like the few that I've talked about, trades, small-time manufacturing, small-time industrial activity, small businesses, it rose by about 3 percentage points. And she also calculated that the employment rate for married women was almost 3 times greater than that registered in censuses, right? So this is pretty path-breaking in terms of showing that your official data is actually undercounting the work done by women, and it's underestimating the work done by women. And this is a theme that kind of resonates even to this day, that the fact that when women's work is invisibilized, then their contributions to the economy are also invisibilized, right? So it's very important for work to be visible, right? To know that they are making very gainful contributions. This is not to say that they are not making gainful contributions as homemakers, but globally, the work of homemakers is not part of our system of national accounts, which means it does not enter the formal GDP calculations, right? The work is very important. In fact, without the work of homemakers, whoever that may be, a man or a woman, it is almost impossible for other people to continue to work, right? If a person is out there working and comes home at night, then he or she is expecting that the house is running, and he or she is expecting that they will be replenished, right? So that's the replenishment of labor, that the depreciation of labor that's being taken care of by people who stay at home. But that work is neither recognized, nor is it considered productive, nor is it part of system of national accounts, and therefore it does not enter our gross domestic product calculations. You have lots of shadow economy and satellite work that's done. So to that extent, although Golden did not frame her work in this kind of what one would call feminist economics argument, but this is what she was showing, that there has been a massive undercounting of women's work, and this is one of the first efforts to look at archival data and bring this to the fore, right? Another key contribution of Golden is that she talked about structural change. Right? So very briefly, for those of us who might have forgotten what structural change is, you probably studied it in your class 10 or class 12 economics and then promptly forgot about it. is that it's a process of labor reallocation across economic sectors, and it has implications for labor productivity. So if you're thinking of, say, the Lewis model, right, which is really about transferring labor, surplus labor from agriculture to the modern industrial sector, right? So that's how an economy grows. That's how productivity increases. So surplus labor from agriculture is absorbed into the modern traditional sector, into the modern sector, and which so labor has a higher productivity, wage rates are higher, and that's one way in which an economy is changing. Right. So the standard model of structural change is that an economy moves from being agrarian to becoming industrialized to a manufacturing stage, and then moves into a service sector economy, right? So labor reallocation from lower productivity sectors to higher productivity sectors is the principal route to improvement in employment conditions in low income economies, where the employment problem is not manifested as underemployment, as unemployment, but it's rather underemployment, right? So you have 5 people doing the job that 2 people could do. So nobody is unemployed, but there is a high level of underemployment, and then there's also, and thereby a prevalence of low productivity work, right? So Golden was also looking at structural change and women's labor market outcomes. Right? So before Golden postulated what became a stylized fact about women's labor market outcomes in economics, is that researchers had said that there is a positive correlation between economic development and women's LFPR. That is, women's LFPR is just rising as the economy grows. So women are far more likely to enter the labor market with economic development. And even a casual look at the statistics of India would show that it's not true. And Golden also found that it wasn't true for US data if you actually look at census records, right? And this partly arose because women's labor market participation had been grossly underestimated in the agricultural and the pre-industrialization era in the US, right? So her historical analysis changed that understanding. She finds that prior to the rise of industrialization in the 19th century, during the period of agriculture, women were far more likely to participate in the labor force. And with the advent of industrialization, it actually became much harder to work from home and combine market work and housework. And therefore, married women's participation actually declined, right? So this graph lays it out very nicely. So what she says is that women's labor force participation follows a U-shaped curve, right? So in the period of agriculture, women were participating in the agricultural sector at very high levels. And this was a period when women's education was low, fertility was very high. And it was also easy. One good thing about agricultural jobs is that they are very flexible, easy to combine agricultural tasks along with housework. Sometimes your farm is adjacent to your house or it's not very far, and you have flexibility options. You can go work, come back, and still take care of the housework, right? And many of them, if they were farm workers, they had to be answerable to an employer. For some of them who were not farm workers but employers themselves, there was greater flexibility as well of work. But with the advent of industrialization, and particularly with blue-collared works, it became very difficult to combine factory jobs, shop floor jobs with housework, right? So, and this was also a period where fertility continued to be high. So there were many children. Home care and child care was fairly demanding, and women's education was still low, right? So it is one of the key aspects of Golden's work is that she points out is that labor market outcomes don't change very fast, right? Because labor market outcomes also depend upon women's expectations of their career, and women's expectations of their career will to a certain extent determine their investment in their human capital outcomes, right? So if parents didn't work, then they might not foresee changes where they think that their girl child would work, and therefore they might not invest in the girl child's education now. So by the time there is a demand for women's labor, there is a lag, so there might be an underinvestment, right? So in any case, what happened with industrialization is that um, there was a retreat of women from the labor market because the job structures had changed. Women could not combine it. Fertility was still high. Their education was still low. And it wasn't also the norm for women to be participating in blue-collared activities, which were the kinds of jobs that the early wave of industrialization threw up, right? Assembly line, working in the shop floor, working as in trading floors as tradespeople, blacksmith, shoesmith, and what have you. All of these were dominated really by men. It wasn't common for women to be seated in these kinds of places, right? And there is also what is called an income effect. Because of the advent of industrialization, household income increased. Women also retreated from the workforce because the man's income was now good enough for them to run their household. However, with increasing industrialization and the advent of white-collar jobs and the beginnings of the service industry, what you find is that, and also aided by the contraceptive pill in 1950s, you find that women's um, women's human capital investments increased. They started to be more educated. Um, Clerical jobs of the kinds that women could participate in were opening up, and family size had fallen thanks to the advent of the contraceptive pill, right? So 1950s is when the contraceptive pill was introduced, and it actually revolutionized women's careers and their expectations of what they could do with their future careers, and it also revolutionized their own educational prospects, right? So with the greater service society, you find that women's um, labor force participation is rising again, right? They are entering the labor market. So this is what we would call also a substitution effect, because with increased human capital investment, the opportunity cost for women to stay at home became very high, which what that means is essentially they were giving up fairly good wages to stay at home. So if they had been employed, if they had put their education to use in the labor market, then they would earn a good wage, right? So this is called a substitution effect, which is sort of overshadowing which is trumping the income effect that I had talked of earlier, right? So this is really the U-shaped curve of women's labor force participation, and this is a stylized fact in talking about women's labor force participation. You'll find hundreds of papers where people are trying to find if women's labor force participation is really following such a U shape across countries, and how that might be related to which stage of structural transformation that country's economy might be in, right? So still a very active area of research. Right. So along with her co-authors, I think one of the papers is Golden and Katz, that really looks at the impact of the pill on women's lives and career in richer countries, right? So why is the pill so different to other forms of contraception? Because this is a form of contraception that is controlled by the woman, right? Unlike other methods of contraception, both of which have to be negotiated with the partner, or is controlled by the partner. Essentially, it gave women control over their ability about the timing of when to have children, right? And given the fact that now you could time your fertility choices, you could also now choose careers or education that required years of investment, right? So prior to the pill, sexual intimacy actually led to pregnancy with a very high probability, and thus women married early, right? You would have to remember that with children coming in early, having children out of wedlock was not really an option. So women were marrying early, having lots of children, and which also meant they were not able to pursue careers that required education, and therefore careers that required a lot of investment, like say even economics or medicine or law, etc., where you had to spend 5 to 6 years because you wouldn't know at which point your career would get interrupted, right? So therefore, it was safer for women to be in professions like teaching or nursing, where career interruptions would not really matter to such a great extent, right? So the power of the pill really is about giving them incentives to invest in their human capital, which of course has a huge effect on their ability to participate in the labor market. Another key insight that was provided by Golden, and one that resonates very much in today's time and world, is about the gender earnings gap, right? So by gender earnings gap, what we really mean is that once you even control, often when you say that there is an earnings gap between men and women, the immediate response that you hear, oh, that's because she is not working, she is that there could be she is low skilled, or she has not years of experience and all of it. But when we talk about a gender earnings gap, one can also control for these kinds of issues and then try and see if there is actually an impact of discrimination on, if there is labor market discrimination against women, right? Golden's contribution to has done quite a lot of work on the gender earnings gap, and she finds that it lessened significantly during the Industrial Revolution, when there was a demand for administrative and clerical services. And also you have to recall that during the Industrial Revolution, a lot of worker productivity perhaps did not differ too much, because the earnings gap, the people were paid by piece rate, right? So the number of sort of garments you stitched, or number of piece rate that you finished up, would determine your earnings. So if men and women were sort of having sort of very similar productivity, there would not be a earnings gap. The earnings gap really came into system with the advent of the more modern systems of payment, where you started having monthly pay rather than pay that is tied to what you produce, what you have called a piece rate, right? Of course, you also have a sort of statistical discrimination, or if you think women are not as good as men, then you can have piece rate that is discriminatory against men and women, irrespective of the quality of work, right? So you often have minimum wages that differ between men and women for exactly the same kinds of tasks. That is not unheard of, and that's also true in India, in our own country, right? So to come back to what Claudia Golden was finding, is that the gender wage gap lessened significantly during Industrial Revolution, but despite economic growth and increasing levels of education among women and a doubling of the proportion of women working for pay, the earnings gap essentially stayed the same between 1930 and 80, right? There was some progress made in the 1990s and 80s and 90s. It was similar, the gap was similar until the 1980s. Then post 1982, if this is far more recent data, you'll find that progress was pretty steep from the 80s and the 90s, but then it seems to have plateaued, right? You find that it was what women were earning, median hourly earnings in the US, as a percentage of men's median hourly earnings was about 80% in 2002. It sort of rose. There's few squiggles out here. There are peaks and declines, and then it's kind of plateaued. But it is plateaued between 80 and 82. It's fairly constant even in 2022. And you find that what is interesting here is that these three lines actually show us that as women age, their pay relative to the pay of men of the same age is declining, right? So if you look at the bottommost, it's the pay gap for women aged 25 to 34 in 2000, and who are going to be 47 to 56 in 2022, right? So in 2000, when they were between 25 and 34, they were earning 86% of what men were earning. But by the time 2022 comes and they are older, in the 47 to 56 age gap, they are earning only 82%. So there's been a fall by about 4 percentage points. And if we look at the top, it's about the youngest age cohort, which is earning about 92. They were 25 to 34 in 2010. So we are looking at younger cohorts of women. When they reach around 37 to 46 in 2022, again, there is a decline of about 8 percentage points. So there is a decline. Essentially, what this graph is saying is that as women are getting older, we see that their pay relative to the pay of men is decreasing, or that the earnings gap is actually increasing. And just some more data which gives you an idea of what's happening. So this is telling us that younger mothers are less active in the workplace than women without kids, and fathers are actually more active, right? So there's a difference. So women with no children are marginally more active in the workplace, whereas men with children are marginally more active in the workplace, right? So there is possibly some story here, right? Similar, that is the extensive margin, which is telling you the proportion of men and women participating in the workplace. And this is the intensive margin, which tells you the hours of work that men and women are able to devote to the workplace, right? So again, you see a similar pattern. No child, women are able to give few more hours, and no child, this is actually closer. It's not making much of a difference in terms of the hours that they are able to devote to the workplace, right? So what is explaining that this residual gap in earnings? And recall, all of this is still looking at US data. So we are looking at a developed country data. We're not looking at anything else at this point, right? So what explains this residual gap in earnings? Is it discrimination, that employers just don't like women and therefore they will pay them a lower wage rate? Is it women's lower ability to negotiate? We all know that starting salaries matter. So if you haven't negotiated well in your first job or in your second job, or when you've made that lateral move, and therefore it stacks up throughout your career, women choose jobs that are less competitive, they are less ambitious, less aggressive, and therefore that's what you're finding picking up. And then perceptions by employers regarding women, right? Slightly different from discrimination. It's just that employers might think that women are not as productive as men, or women are not as committed to the workplace as men are. Women are probably more committed to the home place. It's fairly lot of stereotypes coming in the sense a woman has a child, then they think, okay, well, she's had her child, she's had her maternity leave, now she's not going to be so committed to the workplace, so let's put her on projects that are less challenging, less rewarding. So your career trajectory is beginning to look a little different. Okay? Are these some of the reasons? Then why does it impact time adjusted earnings differentially across occupation, and why does the gap increase with age? So these are some of the questions that Golden investigated through her papers beginning from about 2005, and there were some seminal publications around 2010, work that she has published since then. Right. So what she talks about is what is called a parenthood effect. And I think this graph illustrates that really nicely, that men and women are fairly similar in their career trajectory and in their pay until a certain point in time. And then with the advent of the first child, women's labor market participation and earnings takes a hit, from which it never really recovers, whereas men's career trajectory is unhampered, right? So this is what she called the parenthood effect, and this has really a lot of work has been done, particularly in the developed countries, because you need very good data. Some developing countries have this kind of data, but many countries don't, including India, right? So if you want to try and understand this effect, what you need is probably a lot of women. You need a formal economy so that you can look at administrative data, you can look at tax records, or you need really good panel data where you're following people over their life cycle, right? You can't compare women with no children and women with children, because it's like comparing apples and oranges to some extent. It's never very clear what exactly is happening. But if you are following people over a period of time, over say 30 years, 40 years, then you can see how various events are actually impacting work, how life cycle events are actually intervening with labor market outcomes, right? And that's the kind of data that's being used in many of the papers where from the developed countries that are trying to understand the parenthood effect. right? So I just want to show you a few pictures from this paper that has just been released in August 2023. So Clevin et al. Actually work with, I mean, their original papers were looking at data from the Scandinavian countries, mainly Denmark, and they show a very sharp decline in women's earnings post the event of the first childbirth, right? So here what they present to us is what is called a child penalty atlas for the world, where again they've sort of culled data from very many sources. And so what this is showing is that there is a child penalty in these parts of the world. So the child penalty as a percentage that explains, so the child penalty which explains the earning gap. So this is saying that these darker numbers are saying that a higher proportion is being so the darker portions of the continents are showing that a higher proportion of the earnings gap is actually explained by child penalty or explained by what we call a motherhood effect, right? So the minute women become mothers, they see a huge drop in their labor market participation and in their ability to earn. So you find that these are darker in certain parts of the world, large penalties in Latin America, small penalties in Central Africa, and there is heterogeneity in Europe and Asia. Child penalty is a bit higher in some portions of the Western world, not very high in US and Canada, right? And then what this is showing is that a gender gap in earnings can be explained by a whole host of factors, not just child penalty. There could be labor market discrimination, there could be institutional discrimination, there could be other forms of, there could be other constraints which prevent women from being in the, from being in the market and from earning equally with men. So this is actually now telling you what fraction of overall gender gap in earnings is explained by child penalties, right? So if a large portion of the gender gap in earnings is explained by child penalties, means that the other constraints that there aren't many other factors that are causing a gap in child penalties, right? In earnings, which means that there is no institutional discrimination. You've managed to narrow that. You've managed to achieve equal pay for equal remuneration for equal work, right? Which essentially says that men and women doing very similar work ought to be paid similarly. So the only factor that is standing between equalization of gender gap in earnings is really the child penalty. So the darker colors here signify that above 98.2% or above 98% is now being explained by child penalty, which is actually a good thing. So you've overcome a lot of other barriers to gender gap in earnings, and this is kind of the last barrier standing. So you now find that the Scandinavian countries, erstwhile USSR, some of the communist countries, Australia, these are actually now showing darker colors, whereas a country like India, child penalty is actually not a big thing. We have lots of other factors that are still contributing to a gender gap in earnings, and similarly true for many other countries in Latin America. In sub-Saharan Africa, Latin America is sort of better than Africa, Asia, and the MENA region, but still not as good as Western Europe and the Scandinavian countries, right? So what it's showing here is that the relative importance of child penalties is strongest in North America, Europe, and Australia, followed by Latin America. And Latin America actually tops in terms of magnitudes of child penalties. The relative importance of child penalty is weaker, right, than in North America and Europe. Child penalties explain a relatively modest share of overall gender inequality, that is overall gender gap in earnings in South Asia, Iran, Middle East countries, and Bangladesh, India, right? So what is the explanation behind, say, gender gap in earnings, particularly in the, well, again, to bring home the point that we're talking about developed countries, that it's something about occupations, right? So Goulden's hypothesis posits that it's not differences between occupation. So if we actually equalize the number of women in STEM, equalize the number of women in science, equalize the number of women who go to technology and healthcare sectors, we won't see a reduction in the gender earnings gap. It's actually something happening within occupations. And some occupations, as this graph suggests, there is a greater gender pay gap, right? So what this is showing you is that the coefficient on female and occupation, how that is interacted. So you find that this is the male wage and business incomes at 0. So, and if it is falling below, that means in that particular occupation, women are actually making a lower amount. And if it is above, then women in that particular occupation relative to men are making a higher amount. Of course, you find that there's probably just 1 or 2 places where women are even close, where it says that it does not matter whether you're a man or woman, you're making very similar kinds of uh, earnings, right? But you see, this red is businesses, which largely is very negative. The greens, which is the tech sector, the science sector, that are a little closer. Health, we see a few outliers, but businesses almost are fully concentrated in the negative area, and a fairly strong effect, right? So what's it that's happening? So occupations grouped as businesses have the largest negative coefficients, while occupations that are grouped as technology and science have the smallest one. In fact, for the less than 45 years old, some tech and science occupations show positive coefficients, right? And so in 2010, she followed Golden and a co-author followed a group of Chicago Booth Business School MBAs and said, okay, let's see how their earnings trajectory look from the time that they have received the MBA. So they were followed for a period of 15 to 20 years. And here you're looking at men and women who are coming from the same MBA. So you can't say that one person's degree was better than the other person's degree, and which is why they have received differential pay. No, so this is a very similar educational, the same educational qualifications. Men and women have come in through a fairly stringent process, right? So let us see how it looks, what the trend looks like. Right, so here, this is the ratio of male to female annual earnings. So 0 is the year that they have completed MBA, years since receiving an MBA, and this is the 13th year after that. So the blue line shows the trend line for all MBAs, and then this is the line for women MBA with no kids. And then this is the trend line. So you find that the ratio of male to female annual earnings is at 95%. So women are earning fairly at par when they start off. But as they are progressing, as the number of years post MBAs increasing, at the 13th year, the ratio of male to female earnings is almost only 64 cents to a dollar. So women are making 64 cents to a dollar to what men are making. And these are all, recall, women and men with the same educational background, right? Right. So some of the main reasons that they lay out are career interruptions and average weekly work hours, right? That 18% of women work part-time 13 years after graduation, and most of these are self-employed. So what's happening? Why are the number of hours that women are working sort of so drastically reducing? Why are they moving into self-employment? And you also find some interesting statistics that is correlated with the spousal earnings, right? So the largest reduction in employment is for those with top earning husbands. So in the first 2 years after childbirth, a woman whose spouse was in the top earning group was 22% less likely to be working than if her husband was not, right? So the more your spouse earns, the greater the likelihood that a woman is going to step out of the labor market. After 5 years, she was 32% less likely to be working. Women who have high earning husbands but no children put in as many years of employment and hours of work as do those with less prosperous spouses, right? So there's a clear interaction between having children and having a high earning spouse. So what's happening is that one person's salary is presumably sufficient to run the household, and so the other person is stepping down to take up to do the what we would call reproductive tasks, right? So your reproductive tasks are not only about the act of reproduction, but what economists call reproductive tasks is also uh, about home maintenance, about bringing up children, childcare, and elderly care. So they're choosing different forms of specialization, right? Um, so why is this so? So let's kind of look at hours worked and job continuity. So 70 hours a week is not equal to 2 into 35, right? So if I work 70 hours a week, it's not the same as I'm not going to get paid half time as when I work 35 hours. So there is a certain non-linearity in earnings in certain professions. The more hours of work you're willing to put in, the more you're going to sort of be wedded to your work, the greater you're going to have work-related benefits, right? So if you think of certain professions like investment banking, people are burning out when they're 40. That's because they're working 15 hours, 16 hours of work, right? And they're being disproportionately rewarded for always being available on tap. And this is not always possible for women with young children, right? So as this was a cartoon that kind of illustrates this sort of parental, and so who's specializing at home and who's specializing at work, right? So 6 p.m. meeting might not be a problem for one person, but could be a problem for someone whose main responsibility is at home, right? So people have chosen to specialize at work, or they've chosen to specialize at home, right? So, and there is a penalty that you pay in terms of earnings, right? So certain hours are more important in some jobs, and it's usually those jobs that have high wage gaps, right? So their company is generally determining tasks, priorities, and goals in certain kinds of jobs, and these are attributes of ideal workers in these kinds of jobs, right? Workers in these industries, they face steep penalties for interruptions to their careers. So it shows that for lawyers, a year out of the labor force causes almost 8.4% reduction in salary. So in a recent book that summarizes much of her work and sort of brings it together, it's called Careers and Families. Claudia Golden talks about what are called greedy positions and what are called flexible positions. A greedy position is one that makes a lot of demand on your time. And as you see that this is non-linear in earnings, it's not increasing. So your hours of work are increasing, but your earnings are increasing at a faster rate. Whereas in a flexible position, you want to be available for some time, but you also want workplace flexibility, right? So you want to also be available at the home front. Here, hours of work is fairly linear. What you get, your earnings is linear to your hours of work, right? So this difference is what you are giving up. Someone is giving up for not uh, going into the greedy position of work, right? So if you look, and this is an example that's taken from her book, consider two positions offered to Isabel and Lucas. One is flexible, no matter how many hours the employee works per week, the pay per week is the same. The other position is less flexible, as in the workplace is greedy for your time and therefore is willing to remunerate you much more for the inconvenience of being available 24/7, right? This causes a lot of intra-household gender inequality in the sense of the resource distribution between the people who are choosing to, one who's choosing to be on call at home and someone who's choosing to be on call in the office. And so they're making a choice between equality within the house and maximizing family income. It's possible that both of them, two people, could choose to be in the same flexible position and say, okay, both of us will work here, both of us will devote equal hours of work to the house. Home care and to bringing up children as well, right? But that's not what a rational actor does, right? A rational actor will say, well, our family total household income is maximized if one person chooses to specialize in the office and the other person chooses to specialize at home. And more often than not, thanks to division of labor, gendered roles and responsibilities, the woman who chooses the flexible position or chooses to be available at home is always the woman, right? And she says that there are some occupations where this is not true, and she points to the pharmacy sector as one where it is a high income operation but linear in earnings. And it's according to the data that she presents from looking at US administrative data, it's about the most egalitarian, gender egalitarian of all professions, right? And the reason that she gives is that in a drugstore, you no longer have sort of your mom and pop family businesses where the pharmacist has an intimate knowledge of the customer. Client relations don't matter, right? All they're doing is they have certain very specialized knowledges, and they're using that knowledge to administer to fill your prescription, right? If I go to a bank, I might want to meet the same person I met last time because I've explained a lot to this person and the relationship matters. But in a country like the US, if you're kind of familiar with that landscape, you go to a Walmart or a CVS, you don't care who's filling your prescription, right? You just need someone who is qualified using. There is no personal contact necessary. Drugs have become standardized, right? So all of this means is that there is a greater degree of substitutability between employees. Personal touch is not required. Customer satisfaction does not depend upon customer interaction or personalized customer interaction, right? So pharmacists are largely substitutable for each other, right? So this means that you could work 20 hours or you could work 10 hours. Your pay is not going to change. It is not non-linear for the amount of hours that you're putting in, right? So some of this is also outside choice. You find that men are also suffering, excuse me, that men in the legal profession who take on a non-traditional gender role pay a very high price for that behavior, right? So if the workplace penalizes men more than women for taking breaks from work, then it is a wiser financial decision for a mother to take on more caregiving activities in line with gender expectations. So much of the later work of Claudia Golden has also started looking at corporate culture, what should change about the workplace culture in order for us to achieve what she calls a grand convergence in gender roles both at work and at home, right? So that she's like, it's the workplace culture, it's what we want out of employees that is also driving this divergence, right? If you could move towards like a science or a pharmacy kind of model, if you could have workplaces that have only reasonable expectations out of their employees' time, then we would see a greater equality both in the workplace as well as in economic resources, as well as a division of work within the house. Right, so the implications of her work are many. I'm just broadly going to talk about a few things. Importance of historical research and archival analysis to better understand data, and that economic growth alone cannot pull up women's LFPR. When you say economic development and therefore women's empowerment, women would also start entering the labor force. That's not really true. The process of change matter and the roles of institutions matter, and also trying to unpack underlying mechanisms, right? When we talk about a parenthood effect, how can that be tackled? Is it through norms and policies? It is about thinking about workplace culture. What should the state, the government, as well as society and the workplace be doing to think about achieving what Golden would call a grand convergence in moving towards gender equality in labor market outcomes. So that very broadly is a summarization of her work. I just briefly want to take another 5 minutes to set some of this work in the context of India, where women's labor market outcomes, if you are in tune with what's happening, is that there's a huge concern that women's labor force participation in India is very low. So there are certain supply side issues, there are demand side issues, but there are also issues of gender norms that happen here. So for those who are not in the know, so this is just giving you, so you see the bottom two lines are women's workforce participation rate. The blue line is for both rural areas for men and women, and the red lines are for urban areas, right? So workforce participation rate has been low, kind of steady, a little bit of a plateau, a bit of an uptick in rural areas, but women's workplace participation for a country that's enjoyed almost 8 to 9 years, 8 to 9% growth consistently for more than a decade, this is very low indeed, right? Part of it is related to structural change. This is just showing that even if we look at Indian states, many of them are not actually employing women at the level which we would expect given their level of economic development. So this is what we would expect women's labor force participation to look like. And this is where Indian states are consistently lower than comparable developing countries in terms of their female LFPR, right? So this puzzle is not very clearly understood in India. There's lots of papers around it, lots of research around it for people who are looking for active areas of work. This is one. Women's absence reflects both their and their household preferences. Husbands don't want women to work. There's a social stigma around it. There are issues of mobility. There are issues of gender norms that prevent women, and say childcare constraints. You want a job close to home, but you don't get those kinds of jobs. So these are supply issues, right? And then there are also demand issues where we've had a very peculiar structural transformation in India. We've moved from being agriculture to service sector driven economy. We've kind of skipped the manufacturing stage. And the manufacturing stage is what really pulls up women's employment. So if you've looked at other countries like say Bangladesh, Sri Lanka, Vietnam, Cambodia, South Korea, China, most of them have women's employment has really increased because of large scale manufacturing employment. And this is what is needed to pull out low skill workers from agriculture and move them into employment. We've missed that. We haven't had labor intensive manufacturing. We've had growth led by the service industry, which has not been very employment intensive. And the focus of the service industry has been skill intensive employment and high capital intensive employment. So it's not really favored women's employment, right? Again, you see a relationship between husbands' earnings and the probability of women's entering the labor market. This panel shows that as husbands' earning is increasing, women's labor market is participation is declining, right? So kind of like an income effect, probably a norm effect. As you start earning more, you don't really want your wife to be out in the labor market, but there seems to be some sort of a threshold. So beyond 42 or 44,000 rupees a month, women are re-entering the labor market. Maybe these are women who are more educated. I mean, we need more analysis to understand what's happening. And again, this is an impact of mother-in-law's employment status on the probability of being employed, right? So if your mother-in-law was not employed, very likely the daughter-in-law won't be employed. And if the mother-in-law was employed, very likely the daughter-in-law is employed. This is both in rural and urban areas. What this tells us is the importance of norms, right? So there's also a role model effect, there is a generational effect. So where women are used to being employed, where households are used to seeing women being employed, there's a greater possibility that the younger generation will also continue to be employed, right? And there are measurement issues that Claudia Golden talked about very early in her work that applies still today. What I'm going to quickly is, this is the paper. If you want, you can go and take a look at it, but measurement issues still continue today. When an individual reports on behalf of others, you find that they are underreporting, particularly true of women. So if a husband reports on behalf of his wife, he is likely to underreport her economic activity by about 5.7 percentage points, but that's not true when women report about their husbands, when wives report about their husbands, right? So when husbands report about wives' economic activities, they underreport or they undercount her work, but there is no such difference when we see women reporting about their husbands, right? And this is interesting because we've talked about Golden's work looking at undercounting of women. So some of these issues are still very alive. So you'll see where your data is coming from. And there are several reasons for why such underreporting might happen, is that you don't know the full plethora of work that women do. You think she's a homemaker, but like we saw earlier with Golden's work, she's actually helping out. She might be running the shop as a family contributor, which means that she's not getting paid, but she's weeding, she's threshing, she's working on the family farm. And to you, all of that is just what she does as your wife and as a woman in the household, right? But it actually ought to be part and be counted in labor force statistics. So there is a certain amount of underreporting, and that men are confusing domestic duties and unpaid work of women. And there can also be what is called social desirability bias, what respondents believe men's and women's roles ought to be. So when an enumerator approaches them and they talk about, ask you what the man is doing and what the woman is doing, you might actually report what you think they ought to be doing and not what they actually are. So if you think that there's a stigma, if you as a man of the household think there's a stigma to the woman working, then you'll say, oh, she is not doing anything, she's just a homemaker, because you want to keep you want to maintain a certain social image in the eyes of the enumerator, right? And this is really the references. So just to say that Golden's work, although it has been done in the context of developed countries, largely the US, much of it continues to be relevant. And of course, it has spawned a lot of work in other developed countries. But there's much one can also look at and sort of try and develop her work, extend it and adapt it and theoretically also look at more innovative ways of applying it in our own country context, right? These are the references that I have used in that work. With that, I shall conclude. I think I'm already a little over time, so thank you very much.
Thank you very much, ma'am.
Madam, thank you very much. It was a very, very lucid and informative presentation. I'm sure students who have joined for this webinar would have got a lot of take-home message from your talk.
Thank you.
Thank you, madam. And your talk right from motivation to key aspects of Goldwyn's work, then Indian context, then Goldwyn's work as a unifying framework. You spoke on historical data and measurements, structural changes, U-shaped curve, power of differential, gender earning gap, and so on. It was really very interesting and informative. On behalf of KSTA and all the attendees, I am very thankful to you. My teacher, Dr. I. Karunasagar sir, is there. He's a professor and renowned international scientist. Is there with us. Sir, would you like to say anything about the talk, or you would like to add something?
Yeah, thank you very much. Yeah, because it was very interesting analysis. Because I am not in the field of economics, but I found the analysis very nice, and also you presented not only the work of the recipient but also in the Indian context. So I think it was very interesting for all of us. Thank you very much for the excellent presentation.
Thank you so much. Thanks to KSTA for giving me this opportunity to also, I think, read and reflect a little bit on Claudia Golden's work. It was very useful for me as well to do this presentation. So thank you.
Thank you, sir. Now, Dr. Shailaja is there. Madam, would you like to say anything? Dr. Shailaja.
I think they are unable to connect through audio, sir.
Oh, okay. Are there any questions, Umesh?
No, sir. There are no questions in the chat box as of now.
Madam, on behalf of Karnataka Science and Technology Academy, once again, thank you very much for a very more informative talk. KSTA organizes many programs, both online and offline. Our campus is in GKVK Backgate.
Oh, okay.
Yeah, we will be organizing conferences regularly and workshops. If anything related to economics is there, we'll approach you any presentation and please deliver.
My pleasure, sir.
Yes, ma'am.
Thank you very much. Thank you. Bye.
I also thank all the participants who joined on Cisco WebEx and YouTube platform. Monday, we have one more Nobel lecture series talk on physics. Nobel laureate's work will be delivered by Dr. Philip of Raman Research Institute. Please join by 3:30. Thank you.
Thank you.
Thank you very much, ma'am. Thank you, sir.
Thank you. Thank you.
Karunashagar sir, thank you very much.
Yeah, thank you. It's a great pleasure for me to join this.
Bangalore Bandra. Hello.
ಇಲ್ಲ ಬಂದಿಲ್ಲ, ನಾನು ಇನ್ನು ಮಲೇಷ್ಯಲ್ಲಿ ಇದ್ದೇನೆ, ಹಾಗೆ ನಾನು ಜಾಯ್ನ್ ಆಗುವ ಲೇಟ್ ಆಯ್ತು. ಇಲ್ಲಿ ಅಂದ್ರೆ ಟೈಮ್ ಡಿಫರೆನ್ಸ್ ಇದೆ ಅಲ್ಲ, 2.5 hours.
Thank you very much, sir. We are grateful.
Pleasure.
Thank you, Dr. Shailaja madam, and all the participants. Thank you.